The overall goal of this demonstration is to describe a method for detecting high threshold and subthreshold oscillations in pedunculopontine neurons.
This method can help answer questions about, for example, how cells maintain their firing frequency. How do populations maintain their firing frequency? We know that circuits cannot maintain gamma band or 40 to 60 per second oscillations. Therefore, you require intrinsic membrane properties to maintain that kind of firing frequency. Demonstrating the procedure will be Susan Mahaffey and Dr. Stasia Donofrio.
To prepare standard ACSF, first prepare stock solutions A and B and keep them refrigerated at 4 degrees Celsius for up to 2 weeks. Before each experiment, mix 50 milliliters of stock A with 50 milliliters of stock B. Then, bring the mixture to 1 liter with distilled water and oxygenate it with carbogen at room temperature for at least 30 minutes. Transfer to a beaker for stirring and oxygenate. After bubbling, add the calcium chloride. Next, prepare sucrose ACSF by preparing stock solution C. Before each experiment, mix 300 milliliters of stock C with 600 milliliters of distilled water and oxygenate it with carbogen at room temperature for at least 30 minutes. After that, transfer 100 milliliters of sucrose ACSF solution to a clean beaker